I forgot to ask one question, that why the acid stops carbohydrate digestion? I mean, it's a good question, right? The first answer to this question would be that these enzymes that are, responsible for digestion of carbs, they stop, they just stop. And amylase are proteins itself, right? Because they are protein that breaks down complex proteins, right? Now the proteins are, they get super denatured when the pH is around 2. So we need a neutral pH, okay? And once these proteins get denatured by this acidic pH, the active site shape, it changes, right? And so it no longer fits in the right cup hold, So H ions, they break down these hydrogen bonds that are present in these enzymes, and usually enzymes are these proteins, right? So I hope we understand this, right? Other thing is that there is like chemical instability of this starch enzyme complex, right? We have this enzyme that can, that can survive the acidic environment, but this acidic environment called, the proton, that is H plus, it's gonna, sit on the active site of the, of the enzyme, and it's gonna block the normal catalytic reaction. So that's what also is gonna happen. And if I think on the evolutionary basis, I think each enzyme works at the pH of its own environment, okay? For example, pepsin. Pepsin is the enzyme you need to digest proteins, right? So pepsin, protein digesting enzyme in the stomach, it works great at pH of 2, but it stops working when the pH is around 7, right? So this is great for the body, because our blood is usually it's a group of just proteins, right? RBC is a protein, right? And plasma, and everything is made up of protein. Albumin, globulin, everything is protein. Your blood is just a mixture of proteins, right? Now imagine protein can be digested at pH of 7. If they could have, if they can, then you would not have these globulins and albumins floating in the blood, because blood pH is around 7.3 to 7.4, right? So nature just decided that we're gonna let the digestion of protein be around, happen around 2 pH, okay, pH of 2. So that's the reason why we don't have the digestion of protein around pH 7 and the digestion of carbs in around pH of 2. So that's the reason. I hope I have answered your question. Even if it was not your question, I hope we really understand why it happens in certain ways. So let's talk about lipids today. So we have lipids. The digestion of lipids is done via a lot of things. The digestion is done via stomach and intestine. That's the cool thing. Almost very less, digestion of lipids is done in lingual lipases, that is in the saliva, right? Lingual lipases, right? So stomach produces gastric lipases, pancreas produces pancreatic lipases, and then we have colipases. That's very important, because without colipases, pancreatic lipases will be useless. And then we have cholesterol esterases. That's very important. This cholesterol esterases, it breaks down the ester bonds and gives us free cholesterol and free fatty acids from these complex triglycerides chain, okay? So our diet has triglyceride chains, right? And our main diet is 90% fat, right? It's like a lot of fat, and these triglycerides, we have to break down these triglycerides and get free fatty acids from them, okay? That's the goal of digestion. And vitamin D, right? Which one? Vitamin A, D, E, and K are fat soluble. All of them are hydrophobic. That's why they are fat soluble, okay? If someone asks you why A, D, E, and K are fat soluble, because all of them, A, D, E, and K, can actually result in their own pathways, because they can, cross this bilipid layer which is present on the cell membrane. So they can, for example, I'm just going to talk about vitamin K. Vitamin K can easily jump inside hepatocytes and then produce these factors, right, for this coagulation cascade, right? So that's why A, D, E, K are intrinsically fat soluble or hydrophobic. It means they do not go well with the water. So we need some sort of carriers for them so that they can travel in blood. So that's another concept. Other thing is that we have these cholesterol esterases, right, esters, and then we have these phospholipids. Okay. Okay, so that's our the goal of digestion, right? So let's start a digestion of lipids. The digestion starts at formation of something called as micelle. Now this micelle, or whatever you want to call it, micelle is formed, right, at jejunum, right? And once it is formed, the digestion of fat starts, right? What really micellin is, I think it's pronounced as micelle or michelle or whatever you want to call it. Michelle literally means a little particle. Micelle or whatever you want to call it. I'm so sorry. So micelle is a little particle. So this is a little particle. And what's cool thing about it is that hydrophobic content, that's the content which is really scared of water, which cannot be dissolved in water, is inside the micelle, and the hydrophilic shell is on the outside, so that this whole structure is hydrophilic in nature, but it is carrying a hydrophobic content inside. So this is micelle formation, right? And this micelle formation happens at jejunum. That's very important, right? Now this hydrophobic content, it means bile salts, right, free fatty acid that's inside, cholesterol that's inside, and fat soluble vitamins like A, D, E, K. All of this content is inside, and hydrophilic shell is on the outside, so that this micelle can travel well in the blood, right? So we understand this now. Okay, afterwards what's going to happen is that the first step is done. Micelle formation happened, right? Now, before going further ahead, we must understand fundamentally what is micelle or micelle, whatever you want to call it. I'm going to call it micelle. So a micelle is a tiny droplet, right? It's around 4 to 7 nanometer, right? I don't know why I remember the size of it, but I just remember it because it's small enough to travel in blood. It carries these cholesterol, monoglycerides, triglycerides, fatty acid, bile salts, right? Now, they are not fat globules. They are water soluble transporter pockets, right? Now, why do we even need? I mean, fat is hydrophobic. They don't dissolve in water. Thus, the fat cannot be dissolved in the lumen. Intestinal lumen is really aqueous in nature. We have a lot of chloride in intestine. That pulls a lot of sodium, and that sodium pulls a lot of water. So that's why intestine is really fluid, and we need fluidity in intestine. And because the intestine is really fluid, we need some sort of transporters for these fat, so that it can stay on lumen and get absorbed. So micelle solves this problem by surrounding the fat with this hydrophilic coat, and this hydrophilic coat is nothing but bile salt, okay, that can move freely in water. Always remember that bile salt is hydrophilic in nature, right? And it's an amphipathic molecule. So one end is a water loving side and another is a water hating side. So this is water hydrophobic end, this is hydrophilic end, and bile salts are hydrophilic because the water loving side is on the outside. So this end, the hydrophilic end, is on the outside, and hydrophobic is in the inside. That's how the micelle is formed, right, like this. Okay, I hope you can imagine this beautiful drawing. So this is how it is done, right? So hydrophobic content is inside, right? Now we are at this place, we are in this intestine, right? We are at jejunum, the micelle is formed, we are inside this micelle, we are with this hydrophobic content, right? Now, how micelle is formed? First, let's try to understand this. So the first step of formation of micelle, it happens via emulsification, okay? So this is emulsification, right? First step is emulsification, and this step happens at duodenum. What is duodenum? Duodenum is just this. So imagine this is your stomach and this is your small intestine. This part is duodenum. Just okay. It's the most proximal part to the intestine, right? So no, sorry. It's the most proximal part to the stomach. Okay. So emulsification in the duodenum happens. What really happening here is that these lipids are hydrophobic. Of course, they are fat, and digestive juices are aqueous, and solution emulsify them, right? And okay, what is emulsification? The emulsification is a process by which these large fat droplets from the stomach, they enter into duodenum, and then we have this bile salt that is coming from gallbladder, and this bile has, hydrophobic and hydrophilic end, right? And then this hydrophobic and hydrophilic end, it breaks these large fat globules into small emulsified droplets, increasing the surface area for this enzyme to act. Okay, so emulsification prevents rejoining of these smaller droplets from merging back into one large fat globules, right? So imagine we have one large fat globules and emulsification happen, and then we have these multiple small fat droplets, so that the enzyme that can coat properly, right? Because on a big fat globule, the enzyme cannot, the surface area is really small, so that's why we need emulsification. Okay, so mechanism is that globule prevents, so mechanism, right, break the big fat globules into smaller ones. The bile salts prevents, the bile salt, it covers these small, droplets. Once bile salt covers this small droplets, the small droplets will not, rejoin with each other, and they will not make this big fat globules. That's very important. So bile salts, they prevent the sticking of these smaller ones, right? So globules prevents the formation of large fat globules. Okay, use action, the action of lipases, because now we have more surface area for these small, granules. Okay. Step 2 is this pancreatic digestion. Okay, so once we have this micelle formation, right, let's still make, we have not made the micelle, right? So the emulsification has happened. Now the big fat globules has become the smaller ones, right? And bile has come and coated itself on these smaller granules or smaller droplets of fat cells, right? Now enzymatic digestion happens. So pancreas, it releases these lipases with, of course, co-lipase, and these two, they come to these small droplets, right? It arrives there, and it's like it acts on these droplets, and it breaks these triglycerides into monoglycerides. Now we have these smaller but still hydrophobic molecules, right, floating in the water, right? So bile cells come and they digest the product, and then hydrophobic tail is inward, hydrophilic head is outward, and then we have the formation of micelle, right? So in short, if I have to tell about the formation of, micelle, what really happened is that we have this big fat, really big fat of globules. So emulsification happened. I'm going to write emulsification. Then we have small droplets of fat, right? And then this pancreatic ligase and co-ligase, they come and they work together. And then we have these triglycerides break down into monoglycerides and free fatty acids. Now we have this even smaller monoglycerides and triglycerides and free fatty, monoglycerides and these free fatty acids, smaller units. And then in the end, the bile salts, right, they come and they, work on them. And once they work on them, they, coat these little droplets, and once they coat, they surround these little droplets. The hydrophobic tail is inward, so that all the fat content is inside, and hydrophilic end is on the outside, so that the absorption from the intestine, because intestine is quite aqueous in nature, so the absorption of these little micelle is possible now, okay, because of the micelle. So absorption into enterocytes is done at jejunum, okay, because you understand, we cannot absorb hydrophobic content inside our intestine, because intestinal mucosa, intestinal epithelium is really aqueous in nature. So we need to have micelles, okay? That's the concept here, right? So the absorption inside the mucosa is passive diffusion, right? So micelle approaches this brush border membrane of jejunum enterocytes. The lipid content, that's the inside of the micelle, that's the fatty acid, monoglyceride, cholesterol, it diffuses out of the micelle and enters inside the cell. So note what I'm trying to say. The micelle itself stays outside and only the content slips inside, okay? This is how it happens. Now once the content is inside the enterocyte, the fatty acids, monoglycerides, are once again recombined with each other, okay? So in the mucosal cells, the low concentration of these fatty acids exist, and micelle has high concentration, so passive diffusion of fatty acid contents happens, right? And then free fatty acids plus monoglycerides, right? So reesterification happens, and this we have these triglycerides. Now, why do we even need these triglycerides inside the enterocytes? Because these free fatty acids can actually damage our enterocytes, so we need to make sure that we need to keep them in a smaller, safer way. We repackage them into bigger positions, bigger packages, because we know that if a molecule is bigger in nature, it's going to be more stable in nature. And if a molecule is going to be really small, it's going to be unstable. The energy, it's going to be really, really, really unstable. So we tend to just bind them together so that this high energy packet of molecule, there's the free fatty acids are joined together and they make triglycerides, and now it's a big chain of big chain of free fatty acid. We call this triglyceride, and this triglyceride is not gonna harm our enterocytes, right? So again, the triglycerides are now transported by chylomicrons, right? And they are packed with these proteins called chylomicrons, right? Chylo means small, right? I think chylo means juice, sap. It's a milky juice, and micron means small, of course. Now this chylomicron is coated by something called as phospholipid. Now again, our body, our body has these phospholipid layer around the cell. So chylomicron makes these triglycerides and fatty acid dissolvable in this membranous phospholipid membrane of our cells, right? So the transportation of lipid is done by chylomicron. And then we have these apoproteins. Now we have three types of main apoprotein: apoprotein B48, apoprotein C11, and then apoprotein E. So 48, 3, 48, 2, and E. Now 48 is required by intestine, right? 11 is required by lipoprotein lipases, and E is uptaken by liver. So E is the most important one if anyone asks you. 48 is for the intestine. And so apoprotein is this identity card that says that, hey, I have visited enterocytes already, and now I am free for you to use me. Okay, so this is like this identity card that says, oh, I have already visited this this center called enterocytes of this intestine. Okay, so that's another concept. That's the concept here. That's why we need apoproteins. They are these identity cards. Only the content of micelle is absorbed because of the gradient, because micelles are high gradient for fat and low gradient here, so diffusion happens, and it happens through water layer, small intestine epithelium, of course. Okay, now once these chylomicrons are formed, what happens later is that these chylomicrons are too large for capillaries. They are big packets, right? So what happened here is that they enter into something called as lacteals, right? These are small lymphatic vessels of villi. Lacteals, amazing name. So what happened after this? These lacteals are connected with these thoracic ducts, and these chylomicron, they travel from lacteals to these thoracic duct, and then from the thoracic duct the chylomicron reaches venous blood, and then eventually from the venous blood it reaches the circulation, and then from circulation they reach to tissue. And this is the whole story of chylomicrons, right? Now, before moving further ahead, we must I think it's a good idea to really understand lacteals better, right? Because lacteals are these, I don't know what they are called, lacteal or lacteal. Lacteal, I think they are called as lacteal. And these lacteal are these tiny lymphatic vessels, and they are present in these villus, right? What are villus? Villus are these finger-like projection in the small intestine. Their main job of lacteals is to absorb dietary fats. That's why God has given them. And it's so amazing that how nature has just given us this one channel, this one lacteal opening just for the fat absorption. It's like the maker knew that you will have this problem, and he solved this problem by giving you an extra channel or extra opening. And it's so amazing how it works. It drives my mind crazy how it happens. So why we need lacteals? Why not blood capillaries? Because these chylomicrons are really, really, really bigger in these capillaries. They cannot go directly into the venous blood, because after the fat digestion, the micelles, right? Okay, I'm gonna say something so that you understand this. So fat converts into micelles, right? and these missiles, they enter into intestinal cells, right? And this enterocytes, they convert these fats into these triglycerides, and then triglycerides come into this chylomicrons, and these chylomicrons are these phospholipid and apoproteins, right? So phospholipids and apoprotein means they have phospho, and they have these lipids, right? Fat, and then protein, right? They are large protein particles, basically chylomicrons, right? And these chylomicrons are super big in nature, so they cannot, they cannot go. But lacteals are larger than, these capillaries, and they have these porous opening, so they are perfect for absorption of these chylomicrons. So these chylomicrons, these sugar amino acid, right? And these chylomicrons, they can get inside the lacteals, right? Now, other question that really comes up is that why does the fat go? What happens afterwards? Because here I've written that lacteals, they connect to thoracic duct. Now, what about this whole story of lacteals to thoracic duct? I mean, I don't want to stay in dark. So what happened here is that the fat, let's try to understand step by step. So inside villus, we have these chylomicrons, right? And then we absorb these chylomicrons through lacteal. Now, lacteal, they open up into large lymphatic vessels in the intestinal vessels wall, okay? So these lymphatic vessels, they merge and they form one lymphatic trunk, and then this trunk empties itself into a large sac. It's called cisterna chyli. And this cisterna chyli, from there, this lymph is, what is lymph, by the way? Do you guys know? I mean, lymph is this milky whitey because it contains fat. That's why it's called. Why? I mean, it's a basic question. Why nobody asked this? Why lymph is so white? Why do we even need lymph? Everyone's like, oh, we need lymph because it's our lymphocytes. Oh yeah, we need lymph, but why it exists? I mean, why they cannot be in capillaries? Because the particle size of fat is so big that they cannot be adjusted in capillaries. So this trunk that is full of chylomicron right now, and this sac is now, it opens up, and this trunk, it empties into a large collecting sac. It's called cisterna chyli. Let me write that because it's a good thing to write it, I think. We have this cisterna chyli, whatever you want to call it. Chyli, that's, I think, a white name. Okay, so we have this cisterna chyli. There's a bunch of just fat, right? And from these, the lymph flows into the thoracic duct. That's how it happens. Now this lymph is a milky white because it contains fat, and that's why we call that chyle, chyle, right? Now what happens in the thoracic duct? And this is also a good question. What is this thoracic duct? We should know that. We should not just keep it for some other type of video. Oh, we'll study about it later. No, the thoracic duct is one of the largest lymphatic vessels. It's the main drainage highway for all lymphatic system, thoracic duct, okay? And it starts at this cisterna chyli, right? And okay, cool thing is that this cisterna chyli is present at L1, lumbaric 1, and lumbar 2 way, okay? So this is also nice. And so the path, it goes up, by the way, right? So cisterna chyli, right? It's this thoracic duct starts at L1, L2, and it goes upward through the thorax behind the esophagus. I hope you can imagine. And it ends at this jugular vein, right? Around the left subclavian vein, okay? So the lymph, it drains, and the fat, it drains into the venous blood now, okay? Because inside this jugular vein and inside this subclavian vein, okay? So which vein, if someone asks you like which one, oh, we should know. It's like left internal jugular vein and subclavian vein. So this is important, right? Because half information is bad, I think. So venous blood, it goes to this subclavian vein and in left internal jugular vein, right? So, and then the circulation happens, circulation of lymph, that's the white chime, right? So the whole story of this, absorption of fat was that uh, micelles are formed at jejunum, and then it produces this fatty content inside the enterocytes. And once the enterocytes have this fatty content, we have formation of chylomicron. This chylomicron has these apoprotein. Let's talk a little bit about this apoprotein, right? So let's talk about apo. So the big picture, of this apoprotein is the apoprotein or apoprotein, what do you want to call them? These are these addresses or these identification or labels that tell your body where the particle should go. The key that let enzymes or receptors recognize and act on it. That's the key. That's the identity card, right? Now, what are apoprotein fundamentally? What are these? They are just proteins attached to lipoproteins, right? And they determine their metabolic pathway, their metabolic transport, their function. They are not just for decoration. They're like identity card, right? Now we have these three amazing apoprotein, right? And B48, 11, C11, and then E, right? So apoprotein B48, they are found on chylomicrons, right? Of course, chylomicrons, right? And it's an identity card, right? And it is formed in the intestine, right? Cool thing, right? Other cool thing is that C11, let's talk about C11. So C11 also found on chylomicron, but it is also found on VLDL and HDL. HDL is high density lipoprotein. VLDL is very light density lipoprotein. Now, okay, let me try to explain it. Lipoprotein lipase, it's called LPL, allows triglycerides to break down, okay? And this HDL is acquired from blood, okay? So this C11 is a lipoprotein lipase, okay? And this is used for acquiring, for formation of HDL. That's how we get HDL, okay? Let's open a little bit about C11 and E, okay? Because I think it's really important. Let's talk about E a little bit as well right now. Chylomicron remnants, these are very light, density lipoprotein and high density lipoprotein. HDL, LDL, VDL. I think you guys getting confused, because let me just drop a whiteboard and clear this whole confusion. What happened, how it happens, So, okay. So let's talk about ApoB48, okay? I'm gonna speak right away whatever I know, okay? So inside the intestine, we have these triglycerides. These triglycerides are packed inside chylomicron, and we need this ApoB48 for exit of these chylomicrons. This apo is made by these ApoB mRNA, right? And these ApoB protein, right? They make these ApoB14. Now, this is like a frame or like a scaffold that holds this chylomicron together and helps it to exit the intestinal cell. So without ApoB48, we cannot exit, we as chylomicrons cannot push the chylomicron outside the enterocytes. So that's the function of ApoB48. Now imagine you're inside the lymph, imagine you're inside the blood, right? Now chylomicron has somehow entered the lymph via these lacteals, right, via these thoracic duct, and now this chylomicron is in the bloodstream, and it's in the venous blood pool via the subclavian vein and in left internal jugular vein, right? Now once these chylomicrons are in the bloodstream, they will interact with high density lipoprotein, okay? Now, what are these HDL? HDL are the donor of other apoproteins, okay? So from HDL, they will receive C11 and E. So HDL is necessary for the formation of C11 and E, okay? So that's how it happens. So these chylomicrons have initially ApoB48. And then later they will talk to HDL, and HDL will talk to them, and HDL will, form or give this chylomicron C11 and E. Now, once they have C11, it will activate lipoprotein lipase. And if they have E, it helps the liver recognize the remnants, right? So this is how it happens, right? Now, let's talk about C11 again. Now, once the HDL has given this C11 identification card to this chylomicron, the C11 activates lipoprotein lipases. I'm telling this lipoprotein lipases. What the hell is the lipoprotein lipase? Lipoprotein lipases breaks down triglycerides into free fatty acids or glycerols. It breaks down. It's a lipase of lipoprotein, right? Now, once the free fatty acids are free, they can enter inside the muscle for energy or for the adipogenesis, right? For formation of fat, right? So function of C11 in fundamentally is that it's a turn on switch for LPL enzymes. It allows chylomicron to unload its fat inside the muscle and for the formation of adipose tissue, right? So lipoprotein lipases, we need LPL, and for the functioning of LPL we need HDL, and for the functioning of HDL, for the interaction of chylomicron with HDL to happen, we need ApoB48. So that's the whole story, right? And what happens after triglycerides are removed, right? These triglycerides are removed and the chylomicron gets smaller. Now this again, I can call this chylomicron as a remnant chylomicron, right? Now this chylomicron, once it loses its, this triglycerides, it loses the C11 tag as well. Now, once it loses the C11 tag, amazing, it's so amazing how, it loses the C11 tag back to HDL, okay, but keeps the ApoE right now, okay, so it gives the C11 back to HDL and then it keeps the ApoE. I'll tell you why it keeps the ApoE. I'm going to tell you. So the function of ApoE now is like an address tag that binds to ApoE receptors, right? These are LDL receptors, and they are present on the liver cells. So they help the liver to recognize this used up chylomicron, that chylomicron has successfully given the triglycerides, and now this chylomicron is empty. Now there is no use of this chylomicron. Oh, it has a tag E, ApoE. Okay, the liver is like, okay, it has a tag E, it has no C11, it has just E. Okay, I'm gonna use this chylomicron and I'm gonna shut it off. I'm gonna remove this chylomicron from your blood. So the clearance via liver is done via ApoE, right? So liver removes this chylomicron remnant that has ApoE recognition tag. Now inside the liver, these remnant are broken down again into cholesterol and triglycerides. So when someone says that, hey, don't eat HDL, don't eat this high density lipoprotein, they have no idea, how this whole cycle works, how this whole cycle of B48, C11 and E happens. So if we don't have enough HDL in our system, we will not be able to make these ApoC11 and ApoE. And then if we don't have enough HDL, we won't be able to return this ApoC11 as well, right? So we need enough, we need good quantity of HDL in our blood. Okay, so liver removes this chylomicron, right? So the summary of this whole chylomicron shit is that ApoB is needed for the exit. ApoC11 is needed for the function that turns on this LPL enzyme, right? This lipoprotein lipase enzyme that is present on the endothelial cells, right? And once C1 is there, it's like, okay, it allows chylomicron to unload its triglycerides, and LPL later breaks down its triglycerides into free fatty acids and glycerol. So B48 for the exit, C11 for the digestion via LPL, and then E so that liver can recognize this, fat cell and then, convert this chylomicron back into cholesterol. That was amazing trip we just went to. We just went on a journey to understand this whole concept, right? So B is for, B is for exit, 11 is for LPL, and E is for end of the journey. Okay. So that was the end of chylomicron. I hope we understand this chylomicron concept right there. Let's talk about enterohepatic pathway, enterohepatic circulation. So enterohepatic, in short, is that liver makes this bile, and then this bile goes to terminal ileum, right? and this terminal ileum via portal vein, the bile comes back, and it occurs around 6 to 8 times per day. So it reduces forming of new bile, because forming of new bile is a really expensive process. I even calculated the ATP cost estimated for one bile salt is around 20 ATP. That's super expensive, okay, because one ATP is around 7.3 calorie, right, per mole, and liver makes around 2, okay, liver makes around 0.2 to 0.6 new bile per day. So if it recycles this 15 to 20 grams of bile again, right, because we know that, and bile salt will be around, this much bile salt will be there in this 15 to 20 grams. Imagine if there was no recycling. If there was no recycling, around 24,000 joules of energy would be needed for this 15 to 20 grams of bile, same as running a 10 watt LED bulb for 40 minutes or climbing a 10-meter stairs. And we have more bile salts than stars in Milky Galaxy. Wow, amazing observation. So we have this bile now. We keep recirculating because it's expensive to make new bile salts. Okay, short chain fatty acid, SCFAS, right? Example: propionate, butyrate, and acetate. Why we need them? They are formed by colonic flora, the bacteria of your colon. The function is trophic action. It increases the mucosal growth. Second is acid-base balance. I'm going to talk how. It's absorbed in exchange for H+. I'm going to tell how. And it prevents inflammation. I'm going to tell how. All of these things. So first function is the trophic action. It increases mucosal growth of the mucosa. How? This short fatty acid called this butyrate, right? This butyrate, it's an energy source for mucin and tight junction, right? And this, if FCA is missing after antibiotics or starvation, the colonic mucosal atrophy happens. Okay, before that, let's just read about these SCFAs, because I think it's not making sense to you right now. Okay, so free fatty acids with fewer than 6 carbon atoms is called a short chain fatty acid. Now they are super volatile, they are super water soluble, and unlike long chains, they are short, right? And they are volatile in nature. So what happened here is that the bacteria that is present in your colon, it uses this volatile water soluble 6 carbon fatty acid chain, and with the help of complex carbs, right, it forms something called as SCFAs, that is short chain fatty acid, plus gases, right? Now 95% of gases and SCFAs are absorbed by colonocytes back inside, and the 5% is left in your stool, in your shit, right? So that's why mostly your shit smells like shit, because gases and SCFAs. Okay, another thing is that trophic action. Now understand this trophic action system. So butyrate, right? So this butyrate, because it was formed with the help of these complex carbs, they have some property of energy in it, right? So this SCFAs, that is short chain fatty acids, can be used as an energy source for the production of mucin and for the tightening of tight junction. Not tightening, just they can upregulate tight junctions, right? And this is how the trophic action happens. And if the SCFAs are missing, why? Because you just took a big course of antibiotics. Now, after antibiotics, all of the bacterial flora that was present in your anus, in your colon, now it's gone. Now, after this bacteria is gone, you will not be able to make this SCFAs. Once this SCFAs is gone, you will have something called as colonic mucosal atrophy, because this energy source is absent now. Because you took a big antibiotic course, right? Okay, acid-base balance, how it does it? There it is. So acid-base balance. So SCFA is a weak acid in its own nature, right? And if this is a weak acid, it regulates these NaH2O pumps and it reabsorbs this Na, Now, as I told you already that these are weak acid, it means they acidify the colon, okay? They lower the pH, and this helps to inhibit any sort of bad bacteria that are present, and it favors the good one, right? And how this really happens is that they are already present in the colon, right? I'm setting up the background, right? When SCFAs, these short chain fatty acids, they are produced, they increase the H ion production, right? And they lower the luminant pH, makes it more acidic. This acid environment helps in the prevention of inflammation, of course. And in the body, when SCFAs are absorbed into the blood, they are metabolized to carbon dioxide and water, which normally does not disturb the systemic pH. However, under some problems like diarrhea, or maybe some you're not able to absorb this SCFA, loss of SCFA can slightly affect the systemic acid base balance, right? And other cool thing is that here I've written sodium and HCO3 linkage. Okay, here what happened here is that these SCFA, the chain fatty acid, right? Absorption uses something called as Na coupled transport mechanism. Okay, now this helps NaN water, which is a part of ORS, oral rehydration system. It works by mimicking SCFA like reabsorption, right? So what I'm trying to say is that SCFA influences the local acid base balance in the colon. It promotes the acidity that helps to maintain the healthy flora, all right? And it stimulates these sodium absorption, but they only mildly, little bit, affect the systemic acid base balance. Okay, that's how, because SCFAs are absorbed in these sodium H pumps. So that's how it done. Prevents inflammation because SCFA, they inhibit these NFKB cells, they inhibit T differentiation, and they inhibit bacterial translocation in the mucosal lining. So that was lipids and chylomicrons. I love chylomicrons. It's one of my favorite topic because when I first understood how it really happened, how HDL is not really an enemy, I was in a shock. I was so much in awe that, wow, like how it's just amazing. It's just wonderful. So I hope you have a nice day. I hope you understand the whole story of ApoB48, ApoC11, and ApoE and chylomicron, and thoracic duct, cisterna chyli, and at present at L1, L2, and these triglycerides, right?